I am Anina Sengupta, pursuing MSc in Microbiology from Mumbai University, Barasa, Kolkata. And my today's presentation topic is mitigating cannabis contamination, Bagasse as an eco-friendly absorbent for environmental protection. So here are the contents. I'm skipping that. I'm moving to introduction. So, kanamycin is a broad-spectrum antibiotic which is used to treat infections caused by gram-positive and gram-negative bacteria. So, the emergent diet of kanamycin is 0.15 ppm, and kanamycin can build up in human body through the food chain and has a comparatively narrow therapeutic index and is widely used in human and animal medicines, often given as injections or capsules. Next. So here are the side effects of kanamycin. The side effects include agitation, black territories, bloody or cloudy nose, cough, depression, dry mouth, headache, etc. Here is the effect of kanamycin. So kanamycin targets 30S ribosomal subunit, causing a frame shift in every translation. Overuse of the medication or intake of animal products containing kanamycin can lead to its accumulation in the body, which can have serious side effects such as anaphylactic shock, renal damage, and hearing loss, etc. It also has led to many white bacteria possessing resistance clathrin. So to remediate kanamycin effect, there is two methods. One is conventional and another one is alternative. For conventional method, there are several methods like ozonization, photocontamination, PEC, and electrochemical. So, here's the drawback of conventional methods. Organization may produce toxic byproducts that enhance toxicity and inhibit plant growth or cause harm to microbial life. Photocatalysis and PEC produce new compounds that may not have antibiotic activity but may be toxic. The electrochemical advanced oxidation method has transmitted to large-scale operations, even though the high cost of operation is still a challenge. So, in case of alternative method, we can use biosolvents. Biosolvents in the water treatment field, sugarcane bagasse have been used as an effective absorbent in the removal of heavy metals and dyes from contaminated water due to its fast absorption, kinetics, large surface area, and high stability properties. Therefore, bagasse has been used as a natural water absorbent in the removal of toxic metals PB2 plus ions, methylene butadiene, copper, oil, and malachite green, phenol, cationic surfactant, and many other toxic metals and dyes from contaminated water. Bagasse is composed of 35% of cellulose, 25% of hemicellulose, and 22% of lignin. Here is the structure of bagasse. The next. So the objective of our research is, first of all, we need to know the remediation of kanamycin, why it is become important, because contamination of aquatic habitat, then multi-drug resistance of bacterial growth. Then the objective comes by absorption of kanamycin by vaccine to detect bond pattern alteration, removal efficiency analysis, analytical study to preserve environment using agricultural waste. Next. So for our experiment, we need bagasse, kanamycin, distilled water, and then spectrophotometer, FTIR, weight balance, pipette, tips, centrifuge, and falcon tubes. So here's the methodology comes. First of all, we need to process the bagasse. For that, we took bagasse and then for the washing, then hot drying, and then grinding the bagasse. And then antibiotic solution was prepared. For that, in a certain amount of distilled water, 0.005 gram of kanamycin was added, and the solution was prepared. Then the kanamycin solution was distributed in two different compounds. One is consists of only the kanamycin solution and another one is consists of bagacibitate. After that, we kept both the falcon at room temperature for 2 hours, then centrifuge at 700 rpm for 5 minutes, and then separate the soup and the pellet, and then the centrifuge bagasse, which is in the form of pellet, will exercise. Then to categorize the bagasse, we done the FTIR study, and we use bagasse control, which is normal bagasse powder, and another one is the centrifuge bagasse, which is bagasse plus kanamycin. The next. In the next, the removal efficiency of kanamycin to study that we use UV spectrophotometer, and the removal efficiency is also can be calculated by the equation of the initial term and the initial minus the final value of the kanamycin divided by the final value of the kanamycin into 100. Next. So for the here comes the result. First one is the FTIR result, and the second one is the spectrophotometer result. So this is the FTIR study. In this graph, we can see that the bagasse is the bagasse control, and the bag plus cat means the bag plus the kanamycin antibiotic. So in the first graph, which is bagasse, that is prominent that the bagasse is the transmission rate is higher than the bagasse treated. So it means that the bagasse in its natural form is allowing the lightning pass through more than the bagasse which is treated with kanamycin. So next slide, please. Change in the transmission after absorption. The absorption of antibiotic onto the bagasse may alter the optical properties of the material. It can lead to decrease in the transmission and increase in absorption. This change is due to the fact that the antibiotic molecules may absorb light or scatter in a way that the residues overall transmit through the material. Previous one, please. So in this graph, there are certain big points we have found out that the wavelength number wave number 3744. This is typically associated with the stretching vibration of the hydroxide group. It's often seen in the compound containing alcohol or phenol functional group. In the wave number, the big point found in 2360, the peaks in this region are associated with the stretching vibration of the atmospheric carbon dioxide. However, it's nothing that this region is also close to the region where the stretching vibration of in the wave number 1742, this is commonly associated with the stretching vibration of the carbonyl group. Compounds containing ketones, aldehydes, or ester often show peak in this region. In the wave number 1648, this is associated with stretching vibration of carbonyl group in amine groups. It's often seen proteins and peptides. In the wave number 1037, peaks in this region can be associated with CO stretching vibration in the ethers or alcohols. Next. So it is the study of the UV spectrophotometer. The removable efficiency of kanamycin by bagasse is 60% plus minus 10.33 at 60 minutes when dose concentration of bagasse is 0.00 gram per ml. Next. So in case of dose variation, the removal efficiency of the kanamycin by bagasse is 46.4% plus minus 5.7 at 120 minutes, which is 2 hours. When dose concentration of bagasse is 0.005 gram per ml. Next. So, by this study, we can conclude that kanamycin absorption of bagasse was investigated, and the result illustrated that the absorption of antibiotic had high efficiency and removal performance increased with increasing the concentration of the absorption. Thus, it is concluded that bagasse has significant performance in the removal of kanamycin from aquatic solution, and it can be used for further studies of pollutant removal from water and wastewater in the laboratory and industrial scale. Here are the resources. Now, at present, I would like to thank our honorable Professor Dr. Sunita Mondolman for her guidance throughout the work. And I would also like to extend my gratitude towards our respected HOD sir and other faculties of the Department of Biotechnology, Brainway University, Maharashtra, Kolkata. Thank you.
Good evening, everyone. I, Komal Jarmal, presenting here the part of my research topic, green synthesis and characterization of copper nanoparticles from Moringa oleifera flower and assessment of its antibacterial activity. Today, the major issue of disease progression is the increasing resistance of pathogen to the commercial antibiotics, and to overcome the problem is to search for the alternative resources which are eco-friendly. And the best remedy to overcoming the resistance is the use of nature-based product as an antimicrobial agent. In India, the vast numbers of medicinal plants are present, which shows the antimicrobial activity. In in today's my research topic, I selected the Moringa oleifera plant, which is known as the drumstick tree, horseradish tree, or the Sarjana tree, which belong to the Moringaceae family, which is grown in the tropical and subtropical region of the world. It is a miracle tree because all the parts of its plants are utilized in the traditional medicinal formulation as well as in home remedies. Nanotechnology is the most innovative field which is playing important role in the modern research. It have tremendous uses in various fields such as biomedicine, biocatalysts, textiles, agriculture, food, cosmetics, paints, as well as in water purifications. Nanoparticles can be three alternative methods, that is physical, chemical, and biological methods. Green synthesis of nanoparticles by using plant or plant-derived material has achieved a great interest over the microbe or the chemical mediated synthesis because of its cheap, reliable, facile, non-toxic, eco-friendly, as well as single-step process, and it requires as well as less time-consuming properties. So, green synthesis has advantages over the physical, chemical, and microbe-mediated synthesis. I selected copper nanoparticles for the green synthesis because copper has its own antimicrobial properties, and it is easily available, and it is less expensive than the gold, silver, and platinum. As well as from ancient times, we can use copper vessels for the storing water as it is antimicrobial in nature. Copper nanoparticle synthesis has achieved an enormous interest due to their low cost, easy availability, and numerous applications in various fields, such as it can be used as a sensor, catalytic, inkjet, biofertilizers. It can be superconductor, solar cell, as well as it is helpful in the wound dressings, dye degradation, and biocidal properties. Materials and methods. Preparation of powder and flower extract. For the preparation of powder and flower extract, we use the methodology which is given by the scientist Nigo. In this, Moringa oleifera flowers were collected and then thoroughly washed with deionized water and then dried in the oven at 50 degrees Celsius for 2 to 3 days. Then the powder is prepared by using mortar and pestle, and that powder is again utilized for the preparation of extract. Extract is prepared in the deionized water, and from that extract, we proceed for the phytochemical analysis. Chemical analysis were carried out for determining the presence of alkaloids, tannins, flavonoids, steroids, saponins, polyphenols, glycosides, carbohydrates, proteins, and amino acids. These are carried out because these groups are responsible, bioactive groups are responsible for the antimicrobial properties. Biosynthesis of copper nanoparticles. For biosynthesis of copper nanoparticles, 20 ml of copper sulfate solution plus 80 ml of flower extract, which is prepared in the deionized water, were taken, which is stirred at for 3 hours at 60 degrees Celsius. Then, after 3 hours, copper nanoparticles were formed, and that copper nanoparticles were collected by centrifugation and dried in the oven at 60 degrees Celsius. Again, that prepared nanoparticles proceed for the characterization of nanoparticle, which is characterized by using UV photometer, then FTIR, then XRD, and scanning electron microscopy. To check for the whether that nanoparticles has antimicrobial activity or not, the next method is chosen is the antimicrobial activity of synthesized nanoparticle. For that purpose, pathogenic gram-positive and gram-negative organisms were selected. Result and discussion. Here, the result of phytochemical analysis. Different methods are used for detecting the phytochemicals. In that, we get positive results for all the tests except saponin. The result of UV visible spectroscopy, the formation of particles were confirmed by the UV visible spectroscopy. The UV visible absorption spectrum of Moringa oleifera flower extract shows the lambda max at 264.5 nanometer, while the UV visible spectrum of the synthesized nanoparticles, which are reconstituted in the DMSO, that is dimethyl sulfide, which shows the lambda max at 283 nanometer. From this observation, it is concluded that there is a formation of nanoparticles, as well as the copper nanoparticles usually show the absorbance from 280 nanometer to 360 nanometer, while the aqueous solution shows the lambda max at 620 nanometer. Here the result FTIR. Peak observed in the lower energy region at 498.73 is due to the vibration of copper and oxygen that confirm the formation of nanoparticles, while the peak region at 1021 to 868 is due to the stretching of carbon and oxygen vibration, while the bond observed at 1664 shows the presence of carbon-nitrogen bond.
You have 2 minutes left.
Yes, sir. The broad peak in the higher energy region, that is 3616 to 3270, is due to the bending vibration of hydroxyl group. This is the result of X-ray diffraction. From that observation, it is found that the synthesized copper nanoparticles have the size around the 23.66 nanometer. Here is the image of scanning electron microscopy, which shows that the synthesized nanoparticles in triangular shape. Here the result of antibacterial activity, which is carried out against the gram-positive and gram-negative organism. Here the result of antibacterial activity of flower extract. Here, the remarking result is observed that the Bacillus subtilis and Staphylococcus aureus do not show the inhibition, but synthesized nanoparticle shows the zone of inhibition. From that conclusion, in the present investigation, green synthesis of copper nanoparticles by using Moringa oleifera flower extract was successfully obtained, and that synthesized nanoparticle shows excellent antibacterial activity against all the tested human pathogens as compared to the standard antibiotics and the crude flower extract. This finding suggests that the efficacy of green synthesized copper nanoparticles is higher as compared to the individual crude flower extract and the standard antibiotics, the synthesized nanoparticle has potential for medicinal applications as well as in water purification. These are the some references which are used for the research investigations. Thank you.